Yes, what do you have?
Okay, how do you decide the bodily motions that would connote each type of protein or each creature, like it's a coral?
Yeah.
How do you do that?
Okay, so the proteins are kind of more difficult because we don't know what they look like. So I was just like, two things that look slightly similar. Can I have a GMAP and a GMAP? That's BCL2, yes. So they look kind of similar. So they're protein-esque, is like with the curved arms. But I tried to use the levels, so you can see the BCL2 protein is shorter to show that they're different. But the aptasia and hydra and corals are much more interesting. So if I could, Amy, if you could have, so let's get a coral, aptasia, and hydra. Oh, you're a hydra, Jessie. Okay, cool. So you can see here with our coral, this is Sarah Lynn, her fingers aren't moving, and that is on purpose because corals are basically they look like rocks. And so, but they're really colorful. And so they just kind of stand there, but they're firm. And then we have our aptasia. So I work with them most closely, and they kind of float in the water like Amy is doing with her hands. And they have lots of different tentacles. So that's why her foot is up and her arms are out. And then Jessie over there is a hydra, and they're very simple organisms. And so I wanted to make it straight and firm, but they also kind of I haven't seen them moving. I think they're too small for that. But so that's that. Okay, next question, if anybody has one. Yeah, right here.
What are the three proteins that stops the apoptosis?
Yeah, okay, so can I have my triangle right here? And Jessie, you can be back over there. Okay, so they were all BCL2 proteins. There we go. And then we had MCL1, BCLWA, and BCLWB. And we know about them from vertebrate, yes, things with spines. We have a lot of research done on the people, like humans, that this group of proteins prevents apoptosis from occurring. And so that's what we think also happens in aptasia. So, but that's really all we know about them, or all I know about them at least.
So if you have a bunch of those three proteins, it's what it correlates with them.
I like how your brain is going, but I would be careful on, this is a whole question, and I'll talk more about it in just a little bit with the religion part, but we need to be a little careful with scientists trying to fix coral bleaching, yes, by creating a super coral, because when humans have tried to fix environmental problems, it doesn't always end up well, so we need to think about it through. And so if we think that through, it sounds a lot like cancer, because apoptosis, when that, I'm not using my dancers. Okay, ready? Aptasia. Okay, so, and Jessie, you go be an aptasia too. Okay, if we have no apoptosis, Amanda, go touch Amy's knee. Just touch it. Good. Okay, now put your other hand on it and then grow from it. Ooh, that's cancer. Okay, we don't like cancer, it's bad. It happens when bad cells don't die. And so if we had Jessie be back and y'all go stop her from doing apoptosis. If we have no apoptosis, then Amy's probably gonna get cancer. I think. I actually don't know if cancer is a thing in corals. Dr. Poole would know much more, but it's okay. But that is my thinking. So maybe we need to think about it, but I like it. I'll talk about it more. Okay, I saw another hand somewhere. Oh, yes, back there.
After the dinoflagellates leave the coral, is that it? Is it just dead, or?
That's a beautiful question. Okay, go to the very end of coral. So Sarah Lynn was in green, but she's the dinoflagellate. She got kicked out. Jessie over here, she's trying to survive. So she does the very end. She's like, oh, but where's my partner? I have no food. Right? So There is a point, so when the coral is white, like you'll see pictures of like white corals, and that's why Jessie was in white. That is when the coral is still alive. But then, oftentimes, not all the time, actually, Sarah Lynn can actually come back, and the corals can actually reabsorb dinoflagellates sometimes, but it doesn't happen a lot. Usually the coral dies. Yeah, there you go, Jessie. Good job. Okay, I think we have time for one more question. Yes, Elizabeth?
Can you explain why you decided to use yeast in your experiment?
Yes, that's a great question, Elizabeth. Okay, let us go. Amy, can I have a yeast? Great. Okay, so why yeast? You can be flat, it's fine, so you don't fall over. Okay, why yeast? Such a good question. Can I also have Jessie as an Aptasia? And can I get Mandy, can you clean her? Yes, okay. So if you remember why I explained I used Aptasia as a model organism, so there's this term model organism. It's something that's really grows fast and is easy to be reproduced and doesn't die a lot, so that you can like manipulate it and stuff. So yes, Mandy is really good at cleaning. So Aptasia are a model organism because they're really good at staying alive, super hardy, tough. And and yeast are very similar. They grow really fast. So can you lie down on media? You don't have to clean your Mandy, it's fine. They don't need cleaning either, which is great. They just grow in an incubator. So this is Amy on a plate, which looks like this, in an incubator. And like literally 3 to 5 days later, boom. Stand up, Amy. There you go. I didn't know if you were gonna do it. Yeast. Like we have our results so fast. They're kind of like bacteria if you've ever used those in a lab before. They grow really fast and they're really easy to like manipulate. Amanda, can you manipulate her for an experiment? Oh yeah. Look at that. We know so much now. They're like super easy for that. I know it's weird though, because it's like, why yeast? That's so random. But there's this whole protocol that you can buy, and we bought it, so that's what happened. Awesome. Okay, those were great questions. Thank you so much for that. Now, would you join me in thanking these dancers for their performance?